Hello students, now I will begin with the part 2 of gene cloning video. Here we will talk about the techniques used in cloning. So, what you need to do first, right? So, the technique, the first technique what you need to do, that is the DNA isolation, right? First we need to, because if you want to identify the gene, need to isolate the gene and to make it a clone of that gene, first is that to isolate the DNA, right? Then only you can find your desired gene into that fragment. So, we are going to do the DNA isolation. The most commonly used technique in cloning is to isolate and purify plasmid DNA from transformed E. coli culture. This is what if you are going to make a recombinant DNA by using the E. coli DNA. So, in that process we are explaining here. So, in that way we are taking the E. coli cultures, right. This procedure known as mini prep, this process called as the mini prep, requires as little as 1 ml of overnight culture and is performed by alkaline lysis of the cultured cells, right. This is how they are telling how we treat that particular culture. The alkaline solution of NaOH and SDS breaks up the cell walls, releasing the cellular contents into solution. This is followed by neutralization with potassium acetate and then precipitation of the DNA by 95% ethanol. So, this is the procedure by which they isolate that DNA from the bacterial cell, E. coli bacterial cell. Simplified by the use of mini prep columns, simply employing membrane resin technology, right. By that they will do this. A wide selection of kits is now available. Now, this is what I have told you. This is a little lengthy process to add this, to make a solution, to pour into it, and this and that. But nowadays you have lot many kits which are directly available, right, providing a fast, simple and cost effective way for isolating the plasmid DNA, genome DNA and RNA from microbial, fungal, plant and mammalian cells. So, its process is now easy because of the availability of the various kits we have. So, now we will talk about the gel electrophoresis. This is the second technique. What we need is the gel electrophoresis. Gel electrophoresis is the process of separation, right, by using the various what called the charges separation method. They separate the particular part from the other part which you don't want, like that they are separating it. For the separation of micromolecules, DNA molecules of different size can be resolved by Agarose Gel Electrophoresis, whereas proteins are usually separated by polyacrylamide gel electrophoresis. So, remember, now here concentrate and remember two techniques I am talking about. First, if you want to separate the DNA molecules of the different sizes, you need Agarose Gel Electrophoresis. If you need to separate the proteins of different type, then you need page. So, like that two different techniques are being used. This is like very simple instrument, Agarose Gel Electrophoresis. Most of the labs are having this, right. Here cathode anodes are there and one Agarose Gel medium is there where you pour your DNA and then DNA fragments will migrate, right, as per the charges, and then the isolation will be done, separation will be done. For DNA separation, the gel matrix of choice is Agarose, right. What we are taking gel matrix is Agarose. We make the gel by Agarose. Although acrylamide is sometimes used, particularly for large DNA fragments. The DNA sample, example a restriction digest of a mini prep, is loaded in a wells at one end of the agarose gel slab. You can see in the above structure the wells are there. You can see these wells are there here, right. So, they are loading these DNA fragments into the wells. At the end of the agarose gel slab, which is submerged under buffer, when an electric field is applied, now we switch it on and apply an electric field, the DNA fragments in the digest we call it at that solution where you have that all this DNA together. So, in the digest migrate to anode electrode, right, anode side they will move. The smaller the DNA fragment is, the faster it migrates through the agarose gel. So, you have to make a smaller fragments of DNA for the faster migration. Due to the effect of molecular sieving, which we call it as molecular sieving, because sieving will make separate of two different kind of things. Hence, DNA fragments differing in lengths can be separated into discrete bands. A very simple description, you can understand very well that DNA of different length will be differentiated or separated in that way. You can see this black color photo. This is a photo of the separated DNA in the gel electrophoresis, and you got the different bands, and the different bands will talk about the different length of the DNA fragment. Okay. Here I have tried to give you the detail of this whole process, how to do, right, how to take the bacterium, then make the plasmid isolated, entered into the what called inserted into the bacterium DNA, then inserted into the make more copies, then these copies have been taken out for a culture, and then they have utilized for the various purposes. So, you just go through this whole process, so you can understand that a diagrammatic representation of this process. Now, second technique is the polyacrylamide gel electrophoresis, which we call PAGE. This is also the method where we use this method to resolve the protein bands. We separate the different proteins from that whole bunch of proteins, and this can be seen this particular photo, where the different bands will explain the different type of proteins here. Okay. Please go through and read in detail. I am just talking about to introduce these things to you. PAGE is particularly useful for analyzing gene expression. So, I understood, because you are, when you need not to separate your digest by DNA, but the protein, then it means you are studying something on the gene expression, right. So, here it useful for analyzing gene expressions, right. Recombinant proteins produced by a clone may be identified by comparing electrophoretic bands of the cell extract of the clone versus that of the uh, control, as compared to control, right. So, you can see there is a one control where a normal gene is there, and another there is a modified gene is there, right. Both will go for the protein synthesis process, right. So, both will produce the different kind of proteins, and once you separated these two different kind of proteins, you can easily understand that what you have done is succeeded, or it's working, and you are getting that required gene product or not. Like that, it can help you out to understand that by the PAGE. Next method, you might have heard about this Western blot, to confirm the expression of a recombinant protein, right. Here also, in the above PAGE method, you understood the gene products, you separated the gene products, what the expressed after the expression, what proteins have formed. Here you are confirming about the expression, right. The first step is to separate the proteins in an extract of the host cell tissue by a PAGE, correct, that we have done. The resolved protein bands in the gel are transferred to a nitrocellulase membrane by a technique called Western blot. Like that, what you are doing, these resolved protein bands in the gel, now you are transferring into the nitrocellulose membrane, right, which is there in the Western blot, or this called as a Western blot. the protein gel and membrane are sandwiched between filter paper. Now, that instrument provides you the facility to do this. The protein gel and membrane which is there in the Western blot sandwiched between filter paper, right, submerged in tank buffer, then submerged, then put it down under the buffer and subjected to an electric current, then put on the electric current. What will happen? Bands migrating out of the gel are bound into the adjacent membrane. So, bands which are migrating out of the gel, they will bound to the adjacent membrane, right, membrane which is the Western blot. The protein bands on the membrane are subjected to immunological detection. Now, after that, which bands have reached there, you treat them with immunological processes and to understand that these are those proteins, these are those proteins, right. Next method is called Southern blot, right. These are the instrument for the these blots. This is you can see agarose gel, this is the nitrocellulose membrane, right. This is the thick filter paper under which they make it sandwiched. This is the high salt buffer. Okay, so like that these processes performed. In a similar technique for DNA, resolved DNA bands in a agarose gel after electrophoresis can be transferred to a membrane, nitrocellulose or nylon type of, by technique known as Southern transfer blot. Okay. So, you have seen the above. Here a different membrane has been used. So, different membrane provides a different kind of migration. So, the different technique name is there, Southern transfer. This the process in this case is simple diffusion assisted by pulling a high salt buffer through the gel and nitrocellulose membrane. A particular DNA fragment on the membrane can be identified by hybridization. So, this band will be identified as hybridization. Hybridization already I have discussed little bit in the gene mapping process, adding something new into it, that called hybridization. Okay. Now, next process is called colony blot. Colony blot is a variation of Southern blot, right. Means little different variant of Southern blot. Instead of working with resolved DNA bands, one simply use a nitrocellulose membrane to directly transfer bacterial colonies, right. So, here they are not using resolved bands, while they are using direct transfer. Okay. This is the way they do in the simple method of petri dishes. Hybridization. The primary purpose of conducting a blot in most cases is for DNA hybridization, right. One DNA joined with other DNA is called hybridization, right. Whenever you have something else to be added in something, that formation is a hybridization process. With the labeled DNA probe, hybridization is a DNA denaturation renaturation process. The DNA on the nitrocellulose membrane is first denatured and the single stranded DNA then bind to the radio labeled probe if they are complementary. So, like that you can do the probing by the hybridization process, right. You have certain probe and that you can insert into the DNA, then that is also a hybridization process, right. Here you can see inserted DNA. You can see here single enzyme restriction sites. Here restriction enzymes cut into this particular area, and then they are adding the radioactive probe. You can see here they are adding the radioactive probe, and this radioactive probe has been added and then will be read by autoradiography. You can read it through autoradiography. After hybridization, now hybridization done. Now you understood the after these processes the hybridization done. Now what? After hybridization the membrane is exposed to X-ray film followed by the film development. Only bands that are hybridized with the probe appear in the film, right? Maybe the radio labeled probe, so you can easily identify in the picture the comparative band position, and therefore the identity of the band can be traced in the original gel. Okay. Hybridization is one of the technique to enable the identification and isolation of a clone containing the DNA of interest in vast population of clones in a transformation. So, I hope you understood hybridization is one of the technique to enable the identification and isolation of a clone. So, what for they are doing the hybridization? It helps in identification and isolation of a clone containing the DNA of interest in vast population, right. From the huge population you can identify and you can isolate your DNA of interest. Hundreds of clones can be screened in a high throughput manner. High throughput is a word used for by using certain instrumental methods where you can magnify the processes, or you can increase the process manifold. It will be a very high process by which you will get so many numbers at the same time. So that's the way they utilize it. Hundreds of clones can be screened in a high throughput manner for the target clones in a single experiment. This is a conventional technique used for isolating clones containing the genes of interest. So this is a conventional method, right? We have utilized. They are utilizing conventional technique used for isolating clones containing the gene of interest from cDNA or genomic libraries. In many cases, PCR has become a common alternative to hybridization. Nowadays, PCR has been used more, but the technique is available with hybridization as well. Next is the immunological techniques, right? What is happening in the immunological techniques here? Frequently, after cloning the gene of interest, now this is talking about when you have cloned, right? So, cloning done, what you have to do then? One would like to check for expression. Now, we have to check the expression. A common method to detect the gene product using immunological techniques. So, above also you have seen the page is also doing the similar work, but here the techniques are different because of the different requirement. So, here you have immunological techniques. This method requires that here this method has some requirement. Number one, the gene must be properly constructed and inserted for expression because it is the protein targeted for detection. One thing. Second, the protein needs to be isolated and purified from its natural source, which is then used to raise antibodies, right. So, these two because ultimate purpose is you need the antibodies. Nowadays, scientists are using these techniques for coronavirus vaccine, right. They are trying to make those antibodies. This is a method shown by the diagram that how the immunological techniques performed. You can read all these other texts to understand little more. Next is the next process for DNA gene cloning is the DNA sequencing. If you remember in your syllabus, there is a one full topic on DNA sequencing that I will cover later. Here I am just introducing what the DNA sequencing here to be done for the gene cloning. After a gene is isolated, a complete DNA sequence of the DNA molecule is needed if little information exists about the gene, right. If you don't have much information about that particular gene which you have isolated, so what you need to do, first you have to get a complete DNA sequence, right. In case where the amino acid sequence of the gene product is known, the end segment of the isolated gene should still be analyzed. Even if you know the amino acid sequence of the gene, then also you have to perform this. After all, it is necessary to confirm that the isolated gene has the expected sequence. Suppose that you are targeting to a particular sequence that you have identified, this maybe there are certain changes. Suppose that isoleucine protein amino acid have certain modifications. So, you know what the base pairs, what the sequence for the isoleucine. So, you want to look to that particular sequence for isoleucine. Suppose that you have taken that part gene, but there is no that sequence is not available, then that is not of your use. Okay. So, after all, it is necessary to confirm that the isolated gene has the expected sequence. Moreover, the 5' end portion must be sequenced for proper construction and insertion of the gene into a suitable cloning vector. So, here you have a proper sequence. Sequencing is like you know that you know the whole map of the DNA. You have understood in the gene mapping how to do that. So, here the sequence will help you out to do that. Once you know that, it will be suitable to find the correct vector and to introduce into it. Nowadays, DNA is sequenced as denatured double stranded DNA. Sanger's dideoxy chain termination method is the most widely adopted sequencing procedure. You will be getting more detail on it on the sequencing topic. The method begins with the synthesis of a complementary strand of the single stranded DNA using the enzyme E. coli polymerase 1 Klenow fragment. Right here the Klenow fragment has been used. That's why I explained you there. And a mixture of DNTP. So, this mixture can be adenosine, thymine, guanosine, cytosine, right. A DNA primer is also needed for the enzyme to act. So, you need a primer, you need enzyme, right. What enzyme you are using here? E. coli polymerase 1 Klenow fragment. Klenow fragment, you remember that they have the high polymerase activity. In the synthesis process, the nucleotide are added in a sequence complementary to the DNA being sequenced. So, here in this, once you know the exact sequence, then you have a chance to alter it. So, in this synthesis process, the nucleotide are added because you have added the polymerase. So, this polymerase will start adding the nucleotide into it. So, like that it is happening. Okay. So, because of this purpose, you need to have a DNA sequencing properly. Here just one diagram to show you how this process is happening. In DNA synthesis, polymerization of the DNA strands occurs at different speed. Consequently, a heterogeneous mixture of DNA strand of various length is produced. That is representative of all possible sequences. Okay, when you are producing synthesis going on, polymerization is going on, then you will get a heterogeneous mixture of DNA strand. Means all type of bases are joining into it and make a one DNA fragment, and that also the different lengths you will get, right. Depending on which DNA base pair is used, the termination nucleotide will be either A or T or G or C, like that. So, that also you decide that which terminal nucleotide you want. In fact, all 4 reactions are carried out separately, generating 4 groups. So, if you are taking 4 separate sets where in one termination nucleotide is adenine, second thymine, another guanosine, third is cytosine, or in that way you will get the 4 group of sequences. One group with all ending in the DDATP, one group with DDTP, one with GTP, one CTP, like that you will get 4. DNA segments in each group can be separated by polyacrylamide gel electrophoresis. Then after that you go for the gel electrophoresis and separate them. The bands are read from bottom up to 5' to 3' direction. Like that you can read these bands. We are not skilled enough to read these bands. We have to, the biotechnologists are good enough or skilled enough to understand this. But this kind of photos you will get from after that gel electrophoresis methods. Now, very well known PCR. The polymerase chain reaction utilizes the polymerase activity of the enzyme DNA polymerase 1 to amplify a chosen segment of a DNA molecule. So, amplify, this is a very important word to here understand here. Once you understand the amplify, you can understand what the PCR does, right. So, PCR utilizes the polymerase enzyme and then it will amplify the chosen segment of the DNA molecule, means it will make more of its copies in the faster manner. PCR consists of cycle of repeating 3 steps. So, PCR has 3 processes, 3 steps, 3 steps process: denaturation, annealing and polymerization. This is process has been shown how this PCR functions and gives the repeated, once you get one cycle, then you can repeat the cycles and you will get the number of fragments. First process what the PCR performs is the DNA denaturation. The DNA strands are separated by heating. So, what we do, that the heating process is gives the denaturation. Then primer annealing. Primers, approximate 18 bases primer has been used. Complementary to the flanking region are then annealed to the ssDNA strand by cooling the reaction mixture. So, like this way primer has been added, right, in the single stranded DNA by cooling the reaction mixtures. Third is the once the annealed means added one primer at the one end is the process of annealing. Once the annealing done, then we have to go for the polymerase extension. DNA polymerase synthesizes new DNA strands beginning at the primer using the present ssDNA strand as template. You remember the DNA replication process where you need a primer and then you need a polymerase, and this polymerase and primer will give you the another strand of the DNA. The actually similar process is happening in the PCR, right. The cycle is repeated. Each newly synthesized DNA acts as template strands for the succeeding cycle. If you remember that DNA replication process in your mind, you can understand this very clear. Consequently, PCR yields an exponential increase of the DNA segment, right. So, you will get DNA segment multiple cycles. By the end of the n number of cycles, the number of copies of DNA is equal to 2n, right, where n is the number of cycles. The enzyme used in PCR is Stark polymerase 1, isolated from a thermophilic bacterium Thermus aquaticus. Stark polymerase has high polymerase activity, contains number 3 to 5 exonuclease activity and is resistant to denaturation at higher temperature than E. coli enzymes. So, in this benefit we utilize the Thermus aquaticus here. The product obtained from PCR is sufficient for identification and quantification by gel electrophoresis. So, you got the product from the PCR, then you go for the gel electrophoresis and then separate them and study them. Next is the site directed mutagenesis. So, this is another technique used called site directed mutagenesis. The primary objective in protein engineering is to alter. So, alter means either addition, substitution or the deletion one or more amino acid of a protein to effect a change in its function. Suppose that there is a one protein has been formed. The protein is formed by the addition of the amino acid chain, you know very well. And in the amino acid chain, you have a sequence of amino acid A, B, C, D, E, F, G, H, I, J, K, right? But you don't want K, or you don't want B out of it, or you want something into it. So, like that, you can change something into the amino acid. You have added one amino acid extra, you have deleted one amino acid extra and change the sequence. When the sequence has been changed, a different kind of protein is formed, means that protein's function will be changed. Like that what they are doing here. This can be achieved by modifying the corresponding nucleotide of the gene coding for the protein. For example, a change from T to A in the following DNA sequence will result in substitution of the amino acid phenylalanine with tyramine. So like that you can able just a change of a one substitution T to A and you have got the different amino acid, right. So like that these changes can be done. So here the process has been shown clearly how this process is happening. Exonucleases do that, then added into the single stranded DNA, hybridization process, DNA polymerase acts, then do the transformation replication, right, then separation and purification by the electrophoresis method done, and at the end you have got the matured double stranded DNA. Here substitution process has been shown, here deletion process has been shown. One more method is there which we call it as non-radioactive detection methods. A common method to label a DNA is by nick translation or end labeling with radioactive nucleotide. In recent years, non-isotopic labeling has become popular due to increased safety and environmental concerns. Most non-radioactive labeling methods are based on the enzymatic conversion of a chemiluminescent substrate to a stable intermediate compound that decays and emits light. So here they are not actually bringing out the radio labeled method, right. Here you are using a non-radio labeled method. So probing but without radio labeling. So what they do here, they are by enzymatic conversion they make a chemiluminescent substrate. Chemically they make a substrate which is luminescent, means having a little luminescence out of it, right, and convert into a stable intermediate compound. So that will itself emits the light for you. So suppose that process begins and at the intermediate level, suppose that when that compound formed and it emits the light, immediately you can read and you can understand, yes, the process is going on well. So here they have shown the diagram how to do that. So now in this process you have seen that there are various techniques used in a various sequences, and after that you need vectors, right, because if you don't have the vectors of the vehicle you cannot introduce your genes into the host cell. So for introducing that gene which you have already processed by using all those techniques above, now it's ready to introduce into the host cell. So you need a cloning vector. So first we'll see about it. The introduction of a foreign DNA into a host cell in many cases requires the use of a vector. Vectors are DNA molecule used to transfer a gene into a host cell and to provide control elements of replication and expression. So vectors for bacterial cells. First category of vectors, if you are using a bacterial cell as a host, then which type of vectors you are needed. So first we will look into it. First name is that plasmid vectors. They are very widely used vectors, and these vectors have their origin from extra chromosomal circular DNA called plasmid found in certain bacterial cells. So these vectors, they are originated from the extra chromosomal circular DNA called plasmid found in a certain bacterial cells. So from plasmid these vectors have been made. For cloning use, a plasmid vector preferably contains a number of structural elements. What this vector contains? This vector has one characteristic called replication origin, means where the replication will begin. For replication of the vector DNA utilizing the bacterial host system. Cloning sites, it also having cloning sites, and these cloning sites consist of artificially constructed recognition sequences of a number of restriction enzymes. So, you are identifying the cloning sites, and these cloning sites, or you can say the particular sequences you have recognized, and then these recognition sites means are the sites for the restriction enzymes to act. Multiple cloning sites, there is another term called MCS, multiple cloning sites, serves to facilitate the convenient insertion of a foreign DNA. Third is the selectable markers. These are usually antibiotic resistance genes such as ampicillin resistant and in tetracycline resistance genes. So, what is the purpose of these markers is to screen for transformed cells. So, if you want to screen the transformed cells, these markers should be there. So, just by seeing those markers, you can understand, okay, yes, we have got this. So, these are the way these plasmid, this is the structure of the plasmid vectors. You can see ampicillin resistance and tetracycline resistance sites are there. ORI is the origin of replication or the replication origin site. Here MCS site is there, multiple cloning sites. So, like that the vector plasmid vector looks like. One more important information about the plasmid I want to explain because there is a one category of it, high and low copy plasmids. Plasmid can be grouped into high copy or low copy plasmids depending on the number of molecules of the plasmid found in bacterial cell, right. So, low copy refers to 1 to 25 plasmids per cell and high copy refers to 100 copies and more per cell, right. What is the use? What is the benefit? High copy plasmid vectors are the choice if a high yield of recombinant DNA is desired. If you need more recombinant DNA, then go for the high copy plasmids. On the other hand, it may not be desirable to use high copy plasmid vectors if the gene product is expected to cause adverse effect to the host cell. Suppose that if you have produced more gene products and these gene products will kill the host, so that will not be suitable method to be done. So, the PUC plasmid and its derivative can reach very high copy numbers within the host cell, while the PBr322 plasmid and those derived from it maintain at very low copy numbers per cell. So, like that these are the examples of the plasmids. If you want to know a little more about the PUC plasmids, you can read this below paragraph and understand what the PUC plasmid as an example used. Next vector, very important vector is the bacteriophage vectors. So, I told you initially that bacteriophage are one of the very suitable vectors if you want to insert something into the bacterial cell. Another type of vector is derived from bacteriophage that includes bacteriophage lambda and M13. The lambda vectors and its derivatives are used mostly for the construction of cDNA or genomic libraries. M13 vectors were used primarily to obtain single stranded DNA for DNA sequencing before the development of more recent sequencing methods. Here I am not explaining the bacteriophage lambda life cycle. You please go through with this paragraph to see how the bacteriophage once enters into the bacteria, how it survives, what is the life cycle of it. This diagram will help you out to understand the life cycle of phase. Already you have studied actually the phase life cycle in your microbiology classes, but just go through with it. Now the another vector I will talk about that called cosmids. Cosmids are plasmids containing a bacteriophage lambda cos site. Like it's a variable strain or variable type of plasmid, right, which has a bacteriophage lambda cos site. The hybrid structure enables insertion of large DNA fragments. What it provides you? It is providing you the facility to insert a large DNA fragment. The lambda cos site is required for recognition in packaging. So, that is an important feature of the Cosmed. This is the Cosmed, which has a cos site and the phase helps to transfer this. Next is the Plasmids. Vectors have been designed to combine features from filamentous phase and plasmid. These Plasmids allow the propagation of cloned DNA as conventional plasmids, right. So, here it helps to take the cloned DNA like a plasmid it acts, okay. Because this Plasmid has the bacterial plasmid origin of replication and a selectable marker, which can be an ampicillin resistance gene, which enables propagation and selection of the plasmid form. So, you can read little more about the fragments if you want to know. This is what the fragments look like with the ampicillin resistant site. Another are the yeast cloning vectors. So, yeast are also used as a vector here, and they have other advantages over bacterial system for the expression of a complex proteins. The yeast Saccharomyces is a unicellular microorganism, and many manipulations commonly used in bacteria can be readily applied to the yeast. So, here the yeasts are equally effective and equally useful as the bacterial vectors, right. One more is there called the 2-mu circle. Yeast cloning vectors have been developed based on a plasmid called 2-mu circle found in yeast. So, the 2-mu circle is a 6318 base pair in size and present in the nucleus of most Saccharomyces strains at 6200 copies. So, this way, because this is present there in the yeast are utilized for as a vectors. So, we will end up the part 2 of gene cloning method. I hope you understood the different techniques used and the different vectors used for the cloning process. In the part 3 of the gene cloning, we will discuss about the transformation process, right. So, wait for the part 3. Thank you so much for listening this video here.